Hi everyone, this is your Pavanji from PS Academy YT. Today I am going to discuss about the Nobel Prize in Physiology. The Nobel Assembly at Karolinska Institute has decided to award the 2025 Nobel Prize in Physiology or Medicine to the scientist Mary E. Branco, Institute for the Systems Biology, Seattle, USA, Fred Ramstel, Sonoma Biotherapeutics, San Francisco, USA, Simon Sakaguchi, Osaka University, Osaka, Japan, for their discoveries concerning to the peripheral human system. What is this peripheral human system? How they will work? I will explain now. See, there are innumerable microbes will attack our body in daily life, and our immune system will protect from the innumerable microbes. To detect these innumerable microbes which come inside to our body and outside to our body and enter into our body, there are specialized cells known as T cells. These T cells have millions and billions of receptors, and these T cells have these millions and billions of receptors. They bound to the surface structures in our body and fight against these microbes. They fight against these microbes. Some T cells have self identified the cell protein structures. They self identified the protein structures, and bound by their receptors, they are known as self reactivity C cells, because they are self reactively the receptors is attaching to the cells of the protein structure, so they are known as, those cells are known as self reactivity cells. These self-reactivity cells are very harmful. They may cause autoimmune diseases like type 1 diabetes, multiple sclerosis, and rheumatoid arthritis. See how these, from these self-reactivity cells, how our body will escape. See, there is a one mechanism in the early life. In the early life, these T cells was generated in the bone marrow. These T cells was generated in the bone marrow and traveled to the thymus. After traveling to the thymus, they undergo the test. In the test, there is a destruction of these self-reactivity cells, so that our immune system will be active, and we this will protect our protect from the autoimmune diseases. See, this is the only mechanism as believed from the many years was protected the immune system. This is known as self-tolerance, because automatically these cells when move which is synthesized from the bone marrow and travel to the thymus and go under the test. In the test, there is a destruction of self reactivity cells, and these reactivity cells was described destructed, so it is known as self tolerance. The Sakaguchi was interested in this self-reactivity process, and his interest towards this self-reactivity, he conducted the experiment in the mice. In the mice, he removed the thymus. In the mice, he removed the thymus. After the birth of 3 days, he removed the thymus from the mice and injected to the, see, he injected to the overactive immune system. When he injected to this overactive immune system mice, there is a disease of autoimmune diseases in the mice. The mice is diseased. After this, from the healthy mice, from the healthy mice, He injected T cells to the overreactive immune system. The mice is healthy. He was astonished. What is happening in this mice? What are all the cells is transformed? T cells. What are all the important cells are transformed to avoid these autoimmune diseases? So, in search of these cells, he discovered the new cells, that is positive CD25 regulatory T cells. See, this is very important. Discovered positive CD25 regulatory T cells. The cell surface protein which has having C25, there is a depletion of T cells. When the healthy T cells with the CD25, which regulatory T cells, and these CD25 regulatory T cells was important factor. These cells are transformed to the overactive immune system and avoided the autoimmune disorder. Autoimmune disorder. And these inventions was daily approached by the many scientists, and Bronco, another two scientists, namely Bronco and Fred Ramstel. These two scientists experimented on the mice, that is known as scurfy. These scurfy mice, because these scurfy mice having a scaly skin, it is a kind of autoimmune disorders. They made many experiments on these scurfy mice, and they mapped the X chromosome. They mapped the X chromosome. They find exactly at the middle part of the chromosome there is a mutation which causes the autoimmune disorder. In the They mapped the exactly central part of this X chromosome. After mapping, they found 20 genes. They examined 20 genes, gene after gene. At the last 20th gene, there is a mutation in FOXP3 gene. Among these 20 genes, they examined gene by gene. There is a mutation in the FOXP3 gene. This is very important gene where in the mutation for this gene which causes the autoimmune disorder, where mutation is occurred. This autoimmune disorder known as IPEX. See, this IPEX also linked with human variant. In humans also there is an IPEX autoimmune diseases. So they examine the children with the IPEX disorder. They examine the children with IPEX disorder. Say the same result as seen in the humans. There is a mutation in the FOXP3 gene which causes the autoimmune diseases known as IPEX. So they tell the FOXP3 gene is help to control our immune system. To control our immune system. And as we know, Sakaguchi was introduced the self-regulatory system of self-regulatory T cells, and Bronco and Fred Ramstel was discovered the FOXP3 gene to which controls our immune system. Nowadays, clinical trials are going where there is an increasing of the regulatory T cells. For increasing of regulatory T cells, there is a destruction of unwanted cells in our immune system, and they can used in the organ transplantation. And in contrast to this, cancer, in the cancer there is a down regulation of these regulatory T cells, which is responsible for the malignant cells, for the dividing of many cells. So this year Nobel Prize goes to the Fred Ramstel, Sakaguchi and Branco. Thank you. I will next see you in the next video with a more update. Please subscribe to our channel PS Academy YT. Thank you.